Good morning, friends. In the second year for Advanced Plant Physiology paper, in first credit, we are going to study the topic germination. In this credit, we are going to study in detail concept of seed germination, seed viability, dormancy, and methods used for breaking seed dormancy, along with the factors affecting on seed germination. Along with this, physiological changes takes place during seed germination and methods of application of fertilizer before seed sowing we are going to discuss in detail. Before going to study the term seed germination, we must know the seed structure and what is meant by seed. Seed, it is nothing but the fertilized ovule. Depending upon their fertility as well as presence of cotyledons, two types of seeds are there. Dicotyledons, they are with two cotyledons, and with epicotyl, hypocotyl, two cotyledons as well as the seed coat, while monocotyledons, it is with single cotyledons, and reserve food material, it is in the form of endosperm. Then what is germination? It is nothing but the growth of a plant that is contained within the seed, and it results in the formation of a seedling. These are the different steps for formation of seedling. This is nothing but the process of reactivation of metabolic machinery of the seed, and that results into the emergence of radicle and the plumule. This germination commences with firstly uptake of water by imbibition, most of the times by dry seeds, and this is followed by embryo expansion. This first step of uptake of water is triphasic, where in phase first imbibition takes place, where rapid initial uptake of water takes place. Then the next phase, that is a plateau phase, and in third step further increase in water uptake, and this results in embryo axis elongation and breaks through covering layer to complete germination. Again, depending upon the germination is of two types, epigeal and hypogeal. The word epi means above, where the hypocotyl elongates rapidly. Here this is the diagram where hypocotyl elongates rapidly and it reaches upward, pulling the cotyledons. Here you have observed that these cotyledons are pulled upwards and they are above the soil. Okay, whereas hypogeal means below, where cotyledons even after germination remain below the soil. And here these are the some steps that gives you in detail epigeal and hypogeal type of germination. In short, EP means above cotyledons, move above the soil, and hypogeal cotyledons remain below the soil. For study of seed germination, we must know viability of seed. Then what is viability? It is one which is capable of germination under suitable condition. It includes dormant but viable seeds, in which case dormancy must be broken before viability, and this can be measured by germination. This is a rapid estimate to determine whether the seed is alive or dead. Then what is mean by alive or dead? It means embryo is potentially active or inactive. We know that germination is final expression of viability. A potentially active embryo with low vigor or a dormant viable embryo will not germinate normally. In short, viability, it is nothing but the capacity to germinate seed. Then we go for seed dormancy. Dormancy is nothing but block of completion of germination of an intact viable seed under favorable conditions. We know viable means that is with ability to germinate, but this dormancy blocks that completion of germination even with favorable condition. And most of the times, what is the need of seed for its germination? That is water and gaseous exchange. So dormant seeds do not have capacity to germinate in a specific period of time under any combination of normal as well as the physical environmental factors. This is measured by absence of germination. That is, dormancy is nothing but the block or absence of germination. Then how this dormancy is determined? There are major two phases for determination of it. That is, first, which is based on morphological properties, and second, that is based on physiological properties of seed. Depending upon morphological as well as the physiological properties, there are main five types of seed dormancy. First one, that is physiological, morphological, morphophysiological, physical, and combinational seed dormancy. And this classification is given by Buskin and Buskin in 1998, and this again modified in 2004. First, physiological dormancy. This is abundant form and it is found majorly in the seeds of gymnosperms and major angiosperms clades. Again, this physiological dormancy divided into two types, that is physiological deep dormancy, where embryos excised from the seed will produce abnormal seedling or they do not grow. Whereas in physiological deep, non-deep dormancy, embryos excised from these seeds produce normal seedlings. Sometimes there is a need of treatments or growth hormones like gibberellic acid, or sometimes by scarification or after ripening and in dry storage or cold or warm certification results into the breaking of dormancy. The better example for physiological dormancy is amaranthus. This is a leafy vegetable and many of the species are common weeds. So these seeds of amaranthus, they are with thermodormancy, that is they need a high temperature for their germination, while some grasses of arid region they need a period of drying for their germination. Another type, that is morphological dormancy, where the embryos that are underdeveloped, most of the times in terms of size, but differentiated into cotyledons and hypocotyl as well as the radical. These embryos are not dormant, but they need time to grow and germinate. It means they are with a specific period for their germination, and afterwards they get germinates. And the example is Apium graveolens. In third type of morphophysiological dormancy, no doubt this is combination of morphological and physiological dormancy, where physiologically they are with underdeveloped embryos, but again they are with some physiological component in their dormancy. An example is Osmorrhiza as well as the erythronium. Fourth type of dormancy is physical dormancy. It is due to water impermeable layers of palisade cells in the seeds, which controls the water movement, and because of this seeds cannot absorb the water. And to break this type of dormancy, there is a need of mechanical or chemical scarification. Melilotus trigonella, which is from Fabaceae family, commonly we call it as a methi. Then number of members from family Malvaceae, Asteraceae and Leguminosae, they are with physical dormancy. Most of the times they are with a hard seed coat, and that is impermeable to the water. And the fifth type is combinational dormancy, that is with physical as well as physiological dormancy, where water impermeable coats, they are present in physiological dormancy combined with physiological embryo dormancy. And the well-known example of combinational dormancy are geranium and the trifolium. One interesting dormancy is observed under the secondary dormancy in some seeds of mangroves, and these seeds are with viviparous germination. Many of you are familiar with the viviparous germination, where seeds germinate when they still attached to the parent, and this is the adaptation in mangroves for the saline environment. So this is the diagram that gives you how these seeds can germinate. They are still attached to the plant. You go in detail for this uh, YouTube video that how mangrove seeds can germinate. In the next lecture, we go for other parts of this seed germination topic. Thank you.